feel like
Alright, so anybody have any pointers or how to do that? If you were up here, how would you cut it in a cube out of a beet, a round beet?
Personally, I would try to, so like if it, because it's round, I would try to like eyeball about 2 centimeters, a little bit more than that, just to make sure that, since you can cut mold off, but you can't glue it back together. So about 2 centimeters of like of a round off of it, and then cut that round into a square.
So the only thing is that this is like still rounded. You don't see that?
Oh.
Oh.
Not yet.
It's still rounded. So what if we, can I use the ruler?
Mm-hmm.
Okay. If I use the centimeter side, and we said, and we cut it like flat, like that. So now we have a flat piece.
And then like if it, because it's round, so like the middle is the greater surface area, and then like then when you get to the edge is the small surface area, you could technically make that cut into the 1/2 centimeter by 1/2 centimeter cube.
Yeah.
If that helps. And then do like the middle one, the biggest.
Okay, so like, so the biggest is 2 centimeters, right?
Yes.
Okay, so that's 2 centimeters right there. So if we put that over there, we could do 1, 2, and cut it right here. So then it's flat on that side. Two centimeters by 2 centimeters. There we go. So this is our 2 centimeter piece. Where should I put it?
Just set it off to the side for now.
Okay. Should I cut another piece or?
You can use the leftovers if it's big enough, I guess. As long as it's like cube shaped.
Yeah.
I would assume.
Okay. Well, that works out kind of perfect. By 1 centimeter. And then we have to do the 1/2 centimeter, right?
Yeah.
Okay. So all the pieces are cut.
So now place all 3 of the beet cubes into a small beagle.
And for you, because we're recycling the bleach, you can just put it back in the same container of bleach. You can put them right in there.
Wait, this?
Mm-hmm.
And since it already has bleach in it, now you set a timer for 40 minutes.
Okay, who wants to be our timekeeper? Someone set a timer, thank you. Let's put this under here so that we can watch it as it gives it time. All right, so you're good for now. You can come up and do the last part when we
do
yeah, try to, so that you can put them back on. Right here.
Do you want me to keep them like up here?
Yeah, you can leave them up here, that's fine. That way you don't stain your fingers. Cause it's messy. As you
Yeah.
can see.
I kept looking down. I was like, wait, did I like cut my finger or something?
All right, so now this has like a waiting portion, this lab, right? You have to wait for 30 minutes for us to get our results. So basically, now we have to backtrack a little bit. I wanted us to set this up, but you really have no idea what we're doing right now. So what does beets have to do with cell size? What on earth are we doing? Anybody read into it yet?
Are they supposed to be like the cell?
Yes. Our beets are the cell, which is why they have 3 different sizes. We are looking at efficiency and how a cell is efficient or non-efficient based on its size. Okay, what do we know about a cell? Do things have to get into cells?
Yes.
Yep. Do things have to get out of cells?
Yes.
Yep. How does it do that?
Cell membrane.
Through the cell membrane. So the cell membrane is kind of the site of a lot of traffic, right? Things have to go in and out, and sometimes they can't just simply go through the cell membrane, but they have to be transported with proteins and other things to get it go through the cell. So it's a really high traffic area. So a cell, for it to be functioning optimally, it wants to have the most surface area it can. So why doesn't it just grow giant cells then if we want more surface areas?
Well, because then everything would be huge.
Everything would be huge. And what do we do about the distance it's traveling? We've made it really a big cell. Now it's got to travel long distances. So there's like 2 things fighting, right? We want it to be small enough that we have don't have to travel as far to move things in and out of the cell, but we want it to be big enough to have surface area that is adequate for all of its functions. And we also want it to be big enough to hold everything that we need it to hold, right? Well, tends to be a sweet spot when it comes to cells. And so that's what we're looking at. Our beets are our cells. What do you think is going to happen when we put the beets in bleach? And why is that? Because that's what bleach does. So how is the bleach getting into the beet?
Through the cell membrane.
Through lots of cell membranes, right? It's a vegetable. It's made of cell membranes. And so what's happening is that bleach is diffusing into the beet. And what we're going to be able to look at is how good it is at diffusing with the big one compared to the middle one compared to the small one. Okay, and we'll be able to cut into them at the end and see how much of the red is left on the inside compared to how much is now white, because the bleach will make the beet white. Okay, now there's a couple of things that you have to do in the meantime. I gave you in the module, there is on your paper, you guys picked it up. So what I gave you is a explanation of how to calculate surface area to volume ratios, because that is what's important right now after all, is the surface area to its volume. We're comparing how big the cell is to how much surface area there is, okay? So basically calculating surface area is pretty easy. It's just length times width times how many sides. And a cube has how many sides? 6. So it's length times width times 6 for a cube, always, okay? So, because you're calculating area, but then multiplying it by however many sides there are. And then volume is length times width times height, okay? So in the example, it shows you a 3 centimeter cube. Surface area would then be 3 times 3 times 6 sides, or 54 cubic centimeters. And volume would be 3 by 3 by 3 for 27 centimeters cubed. So that gives us 2 different measurements. It gives us surface area, and it gives us volume. And to compare them, we just put them next to each other. So it's 54 to 27 is the surface area to volume ratio. And then you can also simplify those just like you simplify fractions, okay, to get the smallest numbers. So you're going to practice while these are soaking. You're going to practice calculating the surface area to volume ratios of the 4 cubes that are on your paper. And that's what you have the ruler for. You'll measure in centimeters. And then there's also 3 questions down at the bottom that after you do your calculations, I want you to think about and jot down some answers for that, and then we'll discuss it. Sound good? Okay. The 29th, so Monday.
Tuesday.
You're right, that's Tuesday. Thank you. Okay, so down at the beginning, or at the end, there was a couple of questions that it asked you on this lab sheet here, or on the practice surface area to volume ratio sheet. What trend do you notice as the size of cells increases?
They increase in, like, I don't know if that's the right word for it, but like a pattern. Like, if one part of it goes up by this much, then the rest of it also goes up by that much. Like,
Okay.
if you have, like, your square, and, like, the left, like, height goes up by 2, then the right height is gonna go up by 2.
Okay. All right. Anybody else notice anything different? What do you got, Miranda?
As the cell increases, the ratio decreases.
So as the cell got bigger, the ratio decreased. Okay, so that means what for the cell? Less?
Like equal
Yes.
in terms of the volume and surface area.
Okay, so surface area started going down. The cell started getting bigger and bigger. Surface area started getting smaller and smaller, because the bigger that the cell gets, the less surface area it's going to have. That ratio is going to go down. So the cell's not going to be as efficient. Why is it important for living cells to have this perfect sought after ratio? Why is it important for them? What do you think, Lindsay?
I'm sorry, what was the question?
Why is it important for cells to have a good surface area to volume ratio?
Because if it's too big, then materials get across and
Right.
it makes it less efficient.
Right. And why does the cell want to have a lot of surface area? Melanie?
So that they learn these can really run the cell faster and
Right, because the surface area, the plasma membrane is the site of all of that import and export, and the cell wants to have a lot of that in order to be able to import and export enough of the materials fast enough to keep up with its metabolism. Now, over here I drew a picture of a cell. Drew a picture of a cell. Here and here. These have the same volume. Which one has more surface area? The squiggly one, right? This has more surface area because, and think of the surface area as the blue. Look at how much blue there is because you have this convoluted shape, right? So there's all this membrane in here that this round cell does not have, but all these membranes can be put to use for things going in and out of it. So that's one way the cell will try to increase its surface area, is by changing its shape. So anytime you see things like this in a cell or an organelle, that's probably what's important to it. It's got to have a lot of surface area. What organelle does this look like? Mitochondria. So you know when we're looking at mitochondria, there's something about it that is happening across that membrane that's pretty important to the cell, because the cell has created a lot of surface area in there in order for that to happen. Okay, good. So let's look at our beets. Skylar, you want to come up here and look at our beets? So as she's getting ready, what do we expect to see?
More red in the big one
More red in the big one. And why is that?
because it has greater volume.
Right, because it has greater volume in the, there's a spoon. It has greater volume and it had a lot, the bleach had a lot further to go.
Do you want me to use the plate?
No, just
No.
go ahead and use that, put it on the cutting board, because you're gonna cut them in half. Okay, good. Now you're just gonna cut them in half, like kind of the way you measured them. Like, do you remember which side you measured on?
Like that?
Yeah, so cut it across that way. So then if you, yeah. Cool. So on the lab, you guys have, there's a space for you to record these numbers. She's going to measure. It's on page 2 of the lab, towards the bottom. It says cube size and diameter of red pigment. So she now has to measure the diameter of the red pigment.
Like 1.5 on the big one.
So it's 1.5 centimeters.
Yes. On the biggest one. And like on the medium one. It's 1.1 centimeters.
Got bigger?
Oh wait.
Just the red. Are you measuring just the red?
Yeah.
Do you think when you measured it the first time you went this way?
Oh, maybe. Point 7. Oh, 0.7.
0.7 centimeters?
Yes. So tiny. Okay. 0.2.
0.2 centimeters. Perfect. Okay, you can go ahead and leave those there. You can throw your gloves away.
That's cool.
It does look cool. So, what do we notice? We notice that the bleach did diffuse into there. Okay, it did diffuse in. Depending on the size of the cell, more or less of that diffusion took place. So like in the small cell, you can see that it's almost completely white. So that bleach almost went all the way through the whole cell. So you can imagine that the size being small like that for the cell is going to make it easier for them to get waste products out and get nutrients and things in a lot faster method. Okay, so now you're going to take all of this information and you're going to answer a couple more questions. But I wanted to also talk about the very last page, okay, because all through the questions and things are kind of self-explanatory, but I wanted to look at the very last page. This is called a CER, and this is in a graphic organizer form for you to kind of help you as you're going to kind of put your thoughts in order. But basically, a CER is what? Do we know what it's called? Claim, evidence, reasoning. So we have a claim, and our claim should answer the question, does the size of a cell impact its overall efficiency? Okay, so based on what we just saw, based on what we've talked about, do you think the size of a cell impacts its overall efficiency? So your claim will be a statement that answers that question. And then you're going to look at the experimental designs. Constants. IV means what? Independent variable. DV means dependent variable. What are constants?
Things that stay the same.
Things that stay the same. Okay, and then we have quantitative versus qualitative evidence. What is the difference there?
Quantitative is like the amount, right?
Numbers, yeah. Quantitative always is dealing with measurements and number data. So our quantitative evidence would be the measurements we took. And qualitative evidence means what? Those are more your observations, your descriptions in words of what you saw happening, okay? Now, that last bit, the reasoning, that's really the most important part, because that is where you're taking all of that stuff and kind of making a overall statement, okay? Basically, you're saying, because you're basically looking at, and you're going to restate your claim. You're saying, it's clear that this is your claim. Because of this, I know this because of this, and then you state your evidence. Therefore, and then you kind of wrap it up again, that you know that this is happening based on your evidence. So it's basically a long series. It can be a paragraph, it can be a couple of sentences, but you have to support it. Somebody should be able to read your reasoning here and not know about the experiment, but still understand what you're reading, right? Because it should stand on its own out of context. So you should need to give it enough information to do that, to be able to stand up on its own, okay? So you need to talk about all of that evidence and everything you know about cells and what cells need to be able to do in order to stay alive. Okay, so now my suggestion now that we have our data, is to go back to the beginning and go step by step. Make sure you have gone through all of the checkpoints. You've read all of the little blurbs, because all of that is going to go into helping you write a better CER. Okay? Any questions? Alrighty.